The core assumption behind Mars colonization is simple: humans are adaptable, and with enough technology we can live anywhere. But that assumption quietly treats the human body like a device you can relocate, reconfigure, and expect to keep working. The last 60+ years of space medicine were supposed to prove that idea. Instead, they delivered a more uncomfortable payoff. Human biology appears deeply Earth-locked. For decades, engineers have pushed the visible problems: rockets that can travel farther, habitats that can seal, systems that can make food, oxygen, and water. Those challenges are real, and they are measurable. But while hardware advanced, scientists kept watching the one system that cannot be replaced with spare parts: the human body. They studied what happens when the environment changes in ways no human lineage ever evolved to tolerate. Their data did not come from simulations. It came from people. It came from orbital missions and long-duration flights where astronauts lived in microgravity for months at a time, often for 6 months, sometimes closer to a year. And when those astronauts came home, every one of them carried evidence that the body had paid a price. Not a vague feeling of fatigue, but measurable biological damage. Some of it improved after return. Some of it did not fully return to baseline. The pattern has repeated often enough that it stops looking like bad luck and starts looking like a rule. The body can cope for a while, but coping is not the same as adapting. Endurance is not the same as redesign. That is where the Mars story becomes stranger. Mars colonization plans often imply that, given time, humans will adjust biologically to a new world. Yet the strongest evidence available suggests the opposite. Human biology evolved inside one narrow set of conditions, and those conditions are not background scenery. They are inputs. Gravity is an input. Radiation shielding from a thick atmosphere and a global magnetic field is an input. Pressure and the chemistry of breathable air are inputs. Remove or weaken those inputs, and the quiet maintenance work of the body begins to fail in ways that are not always dramatic at first, but are relentless. So ask the unsettling question: if the body shows damage after months in orbit, what happens when the exposure is not a mission, but a lifetime? Mars is not a short visit scenario. It is a permanent exposure scenario. A place where lower gravity is not an experiment, but the default. A place where radiation is not an occasional hazard, but a constant background. A place where every day is lived inside an artificial bubble that must never slip. If Earth is the environment that built us, then what exactly are we becoming when we live somewhere that removes the very signals our biology expects to feel? And this is only the opening problem, because once the body starts changing, the next question is not whether it can survive, but what kind of human returns. That is the thread that runs forward into everything else. The idea that keeps slipping into Mars colonization plans is that lower gravity will feel like relief. Less weight, less strain, easier movement. After all, if the body hurts under heavy loads on Earth, then surely a lighter world should be kinder. But that expectation quietly assumes gravity is only a burden, when in reality it is also a signal. It is the invisible load that tells living tissue what kind of body it must remain. For years, researchers tried to measure what happens when that load is removed. Not just with theory, but with long-duration spaceflight data and careful physiological monitoring during microgravity exposure. They looked at changes in bone, muscle, blood volume, and balance, then compared patterns across missions. They also used partial gravity models on Earth to estimate what a body might do when gravity returns, but not fully. The effort was not about comfort. It was about finding out what the human body requires, even when it is not obvious. The payoff is simple and slightly alarming. Gravity is not optional for survival. It is essential. Mars gravity is about 38% of Earth gravity. A person would feel lighter there, and at first it would seem like freedom. Jump higher, carry more, run with less effort. But that ease is exactly the problem, because ease removes mechanical stress from tissues. And mechanical stress is not damage in the way people imagine. It is information. Bones did not evolve to sit quietly in a vacuum. They evolved to resist constant gravitational loading. Every step on Earth bends bone microscopically. Every landing sends tiny forces through the skeleton. That constant loading is what keeps the system calibrated. Bone cells rely on mechanotransduction, the process where mechanical force is converted into biological signals. When force drops, the message changes. Less force means weaker signals. The body begins to interpret its own structure as unnecessary. In space, astronauts lose bone density at a rate often described as 1 to 2% per month. The weight-bearing bones degrade fastest because they are the most deprived of their normal job. This is not a theoretical hazard. It is a measurable biological response that repeats mission after mission, even with exercise countermeasures. And now comes the unsettling step. Mars gravity is not microgravity, but it may not supply adequate loading either. If the mechanical message remains too faint, bone loss would continue, not forever, but until the body reaches a fragile equilibrium. Equilibrium sounds stable, but stable does not mean usable. A skeleton can reach a steady state that is biologically real and functionally disastrous. A body can adapt downward into a form that survives the environment, yet cannot tolerate accidents, falls, or the simple demands of work. So the question becomes sharper. When people say humans will adapt to Mars, what do they mean by adapt? A body that can exist there, or a body that can live there? And if gravity alone can rewrite the architecture of the skeleton, then what happens when the next invisible input changes too? Last time, gravity looked like the villain, slowly thinning the skeleton until it could no longer carry a life. But it is tempting to soften that fear with a familiar promise. Exercise will fix it. Stronger treadmills, better resistance devices, stricter routines. Bone loss becomes a problem of discipline and equipment, not destiny. That expectation is understandable because it frames the skeleton as a simple structure, like scaffolding. If it weakens, rebuild it. Yet the effort to understand bone loss in space pushed researchers into a deeper place. They did not just measure fractures and calcium. They studied the living interior of bone, the marrow, the tissue that turns a skeleton into a factory. Bones house bone marrow, and bone marrow does not simply sit there waiting for an injury. It continuously produces the blood itself, red blood cells that carry oxygen, white blood cells that fight infection, and platelets that help the body seal wounds. That is not a side function. It is one of the reasons bone exists as a complex organ rather than inert stone. NASA has described this directly in its coverage of the marrow investigation, noting that bone marrow produces red and white blood cells, and that anemia can affect strength and cognitive ability. Now imagine what bone loss really means in that context. If the architecture of bone degrades, the usable space for marrow can change and the marrow environment can shift. The Canadian Space Agency notes that while bone strength loss during missions is well known, experiments on Earth suggested that fat cells in marrow may increase, leaving less room for producing red and white blood cells. It becomes less about whether a femur cracks, and more about whether the body can keep making the cells that prevent everything else from falling apart. This is where the cascade begins. Reduced marrow function can mean reduced blood production. Reduced blood production raises the risk of anemia, which is not just tiredness, but a systemic reduction in oxygen delivery that can narrow what a person can do physically and mentally. NASA reports that changes in red blood cell volume have been documented since early missions and ties marrow results to the broader problem of anemia in spaceflight. And anemia is not the only consequence. Immune response can weaken in space, with research reviews describing immune system dysregulation during spaceflight that persists through long missions. So, injuries change meaning. A small cut that should close cleanly becomes a question. Will platelets respond fast enough? Will immune cells control bacteria before it spreads? Will healing proceed at the usual pace? When infection resistance drops, the environment does not need to be dramatic to become dangerous. It only needs to be closed, stressful, and far from help. And on Mars, that distance is not symbolic. It is literal. No evacuation in hours. No advanced hospital down the street. A colony would be a population living year after year inside the same exposure that drove bone loss in orbit, while depending on bodies that might slowly lose the ability to regenerate blood, fight infection, and heal. The damage would not arrive as one crisis. It would compound quietly over years, with no clear recovery path as long as the environment keeps delivering the same missing signals. If the skeleton is not just a frame, but a life support organ, then the question shifts again. What happens when the very system that makes blood begins to fail, and the colony is forced to live with that decline as normal? It is tempting to believe that the muscle problem has an obvious solution. If gravity goes away, then replace it with exercise. Build stronger machines, prescribe stricter routines, and treat the human body like a system that can be kept in shape through willpower. That expectation has been tested more seriously than most people realize. On the International Space Station, crews follow intensive countermeasure programs built around resistance and aerobic training, because agencies know that microgravity drives rapid deconditioning. NASA describes exercise on the station as essential for helping prevent loss of bone and muscle tissue, and the daily regimen is often described as taking about 2 hours. The effort is not casual fitness. It is a structured attempt to imitate the missing load of Earth. But the payoff is a hard lesson. Exercise cannot replicate constant gravitational loading. Even with modern countermeasures, scientific reviews still describe significant musculoskeletal losses after long missions. And note that station exercise protocols have not fully protected musculoskeletal fitness across spaceflight history. The body responds to the environment it lives in for the other 22 hours of the day, not just the time it spends training. Muscle tissue is metabolically expensive. It burns resources even when it is not moving. And the body is not sentimental about keeping costly tissue it no longer needs. Remove resistance, and the body removes muscle. On Earth, standing is already resistance training. Walking is resistance training. Every time the body catches itself, balances, climbs, carries, or simply stays upright. Muscles in the legs, core, and posture chain are working against a constant downward pull. That pull is so ordinary it becomes invisible, until it is gone. Mars would remove most of that pull. Mars gravity is about 38% of Earth gravity, meaning the resistance from body weight would be about 62% lower. At first, that sounds like an advantage. Work feels easier, movement feels lighter, but lighter also means quieter signals. The legs do not have to push as hard, the core does not have to stabilize as much, the posture muscles do not have to fight the same collapse. The very ease people dream about is the same condition that tells the body to downsize. In microgravity, muscle atrophy can be fast, and it tends to concentrate in the muscles that normally oppose gravity. Reviews of spaceflight effects describe progressive losses in lower limb and anti-gravity muscles during unloading. Even when astronauts train, measurable decrements can remain after long-duration missions, which tells us something uncomfortable. The problem is not that exercise is useless. It is that exercise is a brief substitute for a constant stimulus. Now extend that reality into a colony. Mars would require lifelong, near-perfect exercise compliance, not for performance, but for basic function. NASA can mandate routines for a handful of highly selected astronauts on a mission with constant supervision and a fixed end date. A settlement is different. Children, older adults, sick days, fatigue, stress, equipment failures, injuries, pregnancies, grieving, conflict, boredom. The entire human spectrum. Population-level compliance is not a reasonable assumption, even before the first machine breaks. So the deeper question is not whether a heroic individual can keep training. It is what happens to the average strength of a whole population over years, when gravity itself no longer provides the daily resistance that shaped the human body. If bone loss threatens the frame, and marrow changes threaten the blood, then muscle loss threatens the ability to work, to build, to rescue, to recover. And on Mars, there is no surplus of strength. There is only what the colony can still do tomorrow. The body has already shown that it will rebuild bone for the gravity it lives in, and trim muscle for the resistance it no longer needs. So it is easy to assume the heart is different. A pump is a pump. Blood is blood. Gravity should not matter to a muscle that simply contracts all day. But that expectation ignores what the heart was built to do on Earth. It did not just evolve to move blood through a loop. It evolved to move blood through a loop in a vertical world. Blood must reach the brain above the heart. while the lower body below the heart constantly tries to steal volume and pressure. Veins and valves help prevent pooling in the legs, but the entire system is tuned for one persistent problem: gravity. Researchers have spent decades watching what happens when that problem disappears. In microgravity, fluid shifts toward the head almost immediately, creating the puffy faces astronauts describe, and the sense that the upper body is overfilled. Reviews of fluid regulation in space describe cardiac distension early in flight and rapid plasma volume contraction in microgravity. The body misinterprets this new distribution as fluid overload, and it responds the way it would on Earth if volume truly were excessive. It sheds fluid. This is where the payoff becomes clear. Plasma volume does not stay constant. It drops quickly. Work in space physiology has long described a reduction on the order of 10 to 15% in microgravity. Within days, the body has recalibrated itself for a world where it no longer needs to fight gravity to keep blood in the brain. And the heart adapts as well. Long-duration exposure to microgravity is associated with cardiac remodeling and atrophy, a reshaping and downsizing that makes sense in the new environment, but becomes a liability when gravity returns. The arteries also change, with vascular remodeling and reduced vasoconstrictive reserve described as contributors to post-flight orthostatic intolerance. That is why returning astronauts can struggle to stand, because the system that once pushed blood upward is now optimized for a flatter world. Now consider what happens if there is no return. Mars colonists would not have a temporary phase of adaptation followed by recovery in Earth gravity. The cardiovascular system would be trained permanently in reduced gravity, with less need to maintain plasma volume, less need to preserve the same heart size, and less need for strong vascular tone. Evidence from long-duration missions shows that recovery from cardiovascular changes can be prolonged, and orthostatic intolerance remains a major operational concern after landing. Mars gravity is not microgravity, but it is still a partial gravity world. The heart would be asked to spend years pumping in an environment where the usual vertical challenge is muted. Over time, the most efficient heart would not be the one built for climbing stairs on Earth. It would be the one optimized only for light activity in a lighter world. So, the question becomes quietly frightening. When people imagine life on Mars, they picture lungs breathing new air, boots crushing new soil, hands building new homes. But what if the limiting factor is not what the colony can construct, but what its hearts can sustain? And if the heart is already rewriting itself for a new gravity, what does that mean for the brain it must keep alive, day after day, in a body that is slowly becoming something else? Astronauts have been studied not just as travelers, but as living experiments in altered gravity. Researchers have tracked the vestibular system in the inner ear, the brain networks that interpret balance, and the subtle changes in the eye and optic nerve that appear only after months in orbit. This effort matters because a Mars colony does not just need people who can survive. It needs people who can move safely, operate machinery, and trust what they perceive. The payoff is unsettling. Gravity miscalibration does not merely confuse the senses, it can damage perception, and in some cases damage vision. Inside the inner ear, specialized organs detect gravity and acceleration. On Earth, these signals act like a silent reference line, telling the brain what down means, and allowing the body to stabilize posture and motion. Reviews of vestibular adaptation describe how altered gravity changes otolith loading and forces the brain into sensory-motor adaptation strategies. But this is not like learning a new sport. These systems calibrate early in life, and when gravity changes, the signals become unreliable. In microgravity, reduced otolith input and conflicting sensory cues can produce disorientation, vertigo, nausea, and degraded performance, especially early in exposure. And the most important detail is how adaptation happens. The brain does not truly fix the missing reference. It learns to suppress or reweight the signals, leaning more heavily on vision and other cues to replace what gravity used to provide. That can work in the controlled environment of a spacecraft. But in a settlement, with ladders, sharp tools, vehicles, and emergencies, living in a constant state of sensory compromise is not a minor inconvenience. It is a daily risk. Then there is vision, the sense most people assume is protected. Yet long-duration missions revealed a pattern now known as spaceflight-associated neuro-ocular syndrome. NASA describes it as changes to eyes and vision during spaceflight and links it to microgravity-driven fluid shifts of blood and cerebrospinal fluid toward the head. The hypothesis is not mystical. When fluids move upward and do not drain the way they do on Earth, pressure and fluid dynamics around the brain and eyes can change. Studies describe cerebrospinal fluid effects in the optic nerve sheath and structural eye findings, such as optic disc edema and globe flattening, meaning the shape of the eye itself can change. A recent review in Frontiers reports estimates that visual changes occur in a substantial fraction of long-duration astronauts, with some sources placing incidence far above half in extended missions. Importantly, some findings and refractive shifts can persist for years after return, and may be permanent in some individuals. Now, carry that forward to Mars. If vision changes emerge after months in orbit, and if some of those changes do not fully reverse, then a permanent settlement becomes a different kind of experiment. Mars would not offer the body a return to Earth gravity to restore the normal fluid gradients. It would mean chronic exposure and slow drift. A colony could function while eyesight gradually degrades, until the day it cannot. And that raises a quiet question that will follow into the next part. If the senses that guide movement and the eyes that confirm reality can be altered by gravity and fluid shifts, what happens to the brain that depends on those inputs to stay stable, alert, and sane? All of those problems still feel like they belong to mechanics, to fluids, to signals that might be engineered around with enough effort. Radiation feels different. Radiation is invisible, and that makes it feel negotiable. Shield it, bury habitats, add layers. The expectation is that the same engineering mindset that builds rockets can build protection, so scientists did what they always do when imagination gets ahead of evidence. They measured. NASA sent instruments to Mars that could sit on the surface and quietly count what the planet actually delivers to living tissue. One of the most important was the radiation assessment detector aboard the Curiosity rover, which monitors the natural radiation environment on the Martian surface. Instead of arguing in theory, it watched the sky day after day. The payoff is blunt. Mars radiation exceeds what human biology evolved to tolerate for long periods. On Earth, life is sheltered by a deep atmosphere and a magnetic field that deflects and absorbs much of the charged particle storm that fills space. Mars has no global magnetic field, and its atmosphere is extremely thin, with surface pressure less than 1% of Earth. That means the surface is exposed in a way that Earth rarely is. The radiation assessment detector results translate this exposure into numbers that sound small until they are compared. Curiosity-based estimates reported that an astronaut on the Martian surface would receive on the order of 0.64 to 0.67 millisieverts per day from galactic cosmic rays, depending on the period measured. That is roughly 640 to 670 microsieverts per day, and it is a steady background that does not require a solar storm to be harmful. Now, set that against the world humans were built inside. Earth average background radiation is a few microsieverts per day, varying by location, altitude, and geology. The exact number is not the point, the contrast is. Mars is not slightly higher. It is an order of magnitude different, sustained. And here is the deeper biological trap. DNA repair systems did not evolve to handle space-level radiation as a normal daily condition. They evolved to manage Earth-level damage, where errors are relatively rare and repair can remain accurate. When exposure rises, repair does not just work harder, it risks working worse, because the number of lesions rises and the chance of mistakes rises with it. Reviews of the Martian radiation environment emphasize that radiation on the surface is dominated by galactic cosmic rays and is a persistent hazard for human exploration. For a short mission, damage is a risk. For a colony, damage is a lifestyle. This exposure never stops for colonists, because Mars cannot turn on a magnetosphere, and its atmosphere cannot thicken on command. And unlike muscle, which can be rebuilt, or blood volume, which can be restored, DNA damage accumulates quietly, cell by cell, year by year. So, the question that follows into what comes next is almost unavoidable. If Mars gradually weakens the body with missing gravity, and steadily injures it with radiation that never turns off, what does permanent human life there actually mean? Not for a single explorer, but for generations. After Mars radiation numbers become real, a new confidence often appears. If the planet has no shield, then we will build one. Thicker walls, denser barriers, a habitat that turns a hostile world into a safe interior. The expectation is intuitive because, on Earth, most threats yield when you add enough material between yourself and the danger. So the effort moved from imagination to particle physics. Researchers use particle accelerators and transport simulations to recreate the radiation that matters most in deep space: galactic cosmic rays. These are not just simple photons. Galactic cosmic rays include heavy nuclei, the kind of particles that carry enormous energy and arrive like microscopic bullets. NASA has emphasized that high linear energy transfer charged nuclei and galactic cosmic rays produce large numbers of complex clustered DNA damage, the kind most associated with severe biological consequences. And this is the payoff that changes the story. Some shielding does not simply stop radiation, it transforms it. A heavy nucleus can cross tissue, leaving a dense track of ionization, a line of damage so concentrated that it does not resemble ordinary radiation exposure. Reviews of high atomic number and energy particle biology describe how these densely ionizing tracks produce complex clustered DNA damage that is repaired slowly or can be irreparable. In other words, a single particle does not just nick one place, it creates a local storm of breaks and lesions clustered so tightly that repair systems face a puzzle rather than a simple fix. This matters because DNA repair systems rely on accuracy. When damage becomes clustered, the likelihood of misrepair rises, and misrepair is where long-term biological risk begins to take shape. NASA descriptions of clustered damage emphasize double-strand breaks as among the most physiologically important lesions, and note that track structure and DNA organization shape the complexity of these events. The danger is not only dose, it is the geometry of damage. Now bring in the habitat wall. Spacecraft and habitats often use aluminum as a structural material, but heavy nuclei striking heavy shielding can create secondary radiation, a spray of fragments, neutrons, and lighter ions produced by nuclear interactions. NASA technical documents explicitly note that conventional materials such as aluminum are not ideal for lengthy missions because secondary radiation produced while shielding can contribute substantially to total dose. In other words, adding material can sometimes rearrange the radiation into a form that penetrates and deposits energy in new ways. Hydrogen-rich materials help, because lighter nuclei produce less damaging secondary radiation, and polyethylene and liquid hydrogen have been studied as shielding materials that do not generate the same level of secondary radiation as heavier counterparts. Experiments and simulations comparing hydrogenous-rich composites to aluminum also report advantages for hydrogen-rich materials in shielding performance against space radiation. But help is not the same as solve. The core issue remains that galactic cosmic rays carry such high energy that no practical mass of material can fully block them without creating other problems. So the next idea is to let Mars itself become the shield. If colonists live underground, the regolith above them absorbs part of the radiation. Studies of regolith shielding concepts show that burying habitats can meaningfully reduce exposure, and popular science coverage of modelling work describes substantial reductions with meters of shielding. Yet, even optimistic reductions still leave a background far above Earth, because the source is not a single burst, but a constant rain of particles that keep arriving from every direction. This is the hard conclusion. There is no perfect wall against galactic cosmic rays. There are only trade-offs. Thicker barriers can produce secondary radiation. Lighter materials reduce that effect, but cannot eliminate exposure. Underground living helps, but does not restore Earth conditions. So the question becomes darker and more specific. If the colony cannot build an Earth-like radiation environment, then what does it mean to raise a life in a place where every cell division is happening under a sky that never stops firing? For years, geneticists have tried to quantify how many new mutations humans accumulate under normal Earth conditions, then asked what happens when that baseline changes. One widely cited population genetics review estimates that an average of about 70 new mutations arise in the human diploid genome per generation. Another review of de novo mutation studies places typical counts for single nucleotide variants in the range of roughly 45 to 60 per genome per generation. The exact number varies by method and definition, but the reality is stable. Every generation begins with new copying errors. On Earth, most of these new mutations are neutral. Some are harmful. Natural selection removes harmful mutations, but it does so slowly through reduced survival or fertility, and it cannot inspect every error instantly. Work on mutation load in humans notes that each individual receives new harmful mutations, and that selection removes them through reduced survival or fertility. That is already a difficult balance in a protected environment. Now place that balance under a sky that never turns off. Radiation does not just increase the number of DNA lesions in somatic cells, it can affect germ cells too. A genome-wide mouse study of ionizing radiation found that while baseline de novo single nucleotide variant rate did not necessarily increase significantly, the spectrum of mutations changed, including a significant elevation in clustered de novo mutations in offspring of irradiated fathers. That matters because clustered mutations are precisely the kind of complex damage expected from high-energy particles, the same style of damage discussed for heavy ion exposure. This is where the payoff becomes hard to escape. If Mars increases mutation pressure year after year, then the colony is not just facing medical risk. It is facing an evolutionary math problem. Elevated mutation rates can exceed the capacity of selection to remove deleterious changes, especially in small populations where genetic drift and founder effects already reduce resilience. The mutational load accumulates, and the cost is paid in the most sensitive places first. Fertility declines, not as a single event, but as probabilities shift. Miscarriage rates rise as embryos inherit combinations that cannot complete development. Genetic diseases become more common because rare, harmful variants no longer remain rare. And even subtle harms matter, because a colony does not need dramatic syndromes to collapse. It only needs average function to fall, lower average cognition, lower immune competence, higher developmental instability, higher chronic disease burden. Over generations, population fitness can erode until the settlement becomes a place where survival requires constant medical triage rather than growth. Selection can remove harmful mutations, but it does so by removing people from the future. In a large Earth population, that cost is diffused. In a small Mars colony, it becomes a direct hit to continuity. The very mechanism that could in theory stabilize the gene pool would also shrink the population that must keep the colony running. So the question shifts again, and it becomes almost intimate. A Mars colony is often imagined as a place where children are born under a different sky. But what does it mean to bring a lineage into an environment that may steadily increase the error rate in the code of life itself? And if the damage cannot be fully shielded, then what exactly is being inherited? Generation after generation. Fertilization may still be possible in low gravity, but the earliest steps after fertilization are not just chemistry, they are choreography. The embryo divides and organizes, building a symmetry and structure so that cells know where they belong and what they will become. Simulated microgravity experiments in mice have reported blocked cell accumulation at early stages in embryos exposed to microgravity simulation, suggesting gravity-related cues can matter during specific windows. Work on oocytes also indicates that simulated microgravity can compromise processes involved in cell polarity and asymmetric divisions, mechanisms that are central to early development. Then comes implantation, the moment the embryo must attach and negotiate a relationship with the maternal body. Reviews of reproductive health in spaceflight environments describe that space stressors can impair implantation and placental formation, risking abnormal development. The placenta is not a passive organ. It is a complex interface, and its blood vessel formation is critical for delivering oxygen and nutrients. Broader placental biology literature shows how sensitive pregnancy outcomes are to disrupted vascular development, with vascular defects linked to pathological pregnancies and fetal growth restriction. Even if a pregnancy continued, development would unfold inside a gravity environment that changes nearly every input the fetus expects. Skeletal development is not simply a genetic blueprint. It requires mechanical stimulation. A review on mechanical signaling for bone modelling and remodelling states that proper development of the skeleton in utero and during growth requires mechanical stimulation, and that bone cells translate mechanical energy through mechanotransduction. If mechanical loading is altered for months, bones can form with incorrect architecture, not because genes failed, but because the physical signals that guide growth were muted or distorted. The heart faces a similar logic. Earlier parts showed that adult hearts remodel for the pressure and flow patterns of reduced gravity. A developing heart would be building its structure in that same altered world, with blood distribution and vascular tone different from Earth norms. And the vestibular system, the inner ear reference that anchors balance and orientation, calibrates early, then becomes the foundation for how the brain understands motion and posture. Reviews of vestibular challenges in space emphasize that altered otolith loading forces, adaptation and reweighting of sensory cues. If calibration begins in a permanently altered environment, the wiring may not be wrong for Mars, but it may be incompatible with Earth, and more importantly, vulnerable even on Mars when signals remain incomplete. This is the deeper fear behind generational thinking. Each generation is not just living with the chronic stresses of Mars, it is developing inside them. If early errors accumulate, recovery is not a return flight away, it becomes the baseline. So the question that follows is no longer about whether adults can endure Mars. It is whether a stable human life cycle can exist there at all, from conception to adulthood, without the planet quietly rewriting development into a form that can survive, but cannot truly thrive.